I'm part of Group 6, and we'll be presenting question 2. We'll be describing how the mechanism of UV apoptosis works. The mechanism for generating a low light response is to use a positive feedback loop. The rapid release of CYP-C from mitochondria as a response to a red cell damage could result in such feedback loop. The presence of broad-spectrum capsase inhibitor ZVAD prior to exposure to UV light affected the time of the CYP-C release. The second question: did ZVAD affect the time of CYP-C release? As shown on the graph on the right side, this graph right here, the graph demonstrates that capsase inhibitor did not have a positive effect on cytochrome C release, as the apoptotic cells under stimulus demonstrates no significant difference in time required to release cytochrome C. So the difference between these two there is a difference, but it's not significant enough to determine that that ZVAD does affect it. And then the question, or part 3, or C, I guess, if caspase activation provided feedback loop to site C release, how would the graph shown be different? This is the graph that was on the previous slide. We talked about how there is a difference, but it's minimal. How would it be different? So in order for it to be different, the difference between the two would need to be greater. So the greater difference would have to give more definitive. The greater difference would give more definitive evidence that caspase affects the site C release. The graph on the right shows how the graph would look if there was a greater difference. So this is the original graph, and here's the new graph. As you can see, I had a little block to kind of show that there would be a difference. So the greater difference would just show that the difference is more induced by the change by the caspase. Otherwise, this could just be a randomized result. This could be this way or that way, and you can't really tell. But this gives a more definitive result.
Hello, my name is Deborah Tasu, and today I'll be presenting over the role of RAS protein. This is the first part of question 3. RAS protein acts as a binary switch in signal transduction, altering between the on and off states. In diseases such as cancer, mutation keeps RAS protein steadily active. While the structural biases and controlled pathways are known, the exact mechanisms of RAS function, specifically its interaction with membranes, remains less clear, but crucial for addressing the RAS-related diseases. The figure to your right is labeled as figure 1 in the research paper RAS protein and their regulations in human diseases. And this figure is showing the conformational changes in switch regions when RAS transmission from inactive GDP-bound state to active GTP-bound states. Switch 1 and switch 2 regions are colored in blue and violet, respectively, and the side chain atoms of residue that undergo large conformational changes during transition are shown in stick representations. And interactions formed by the gamma phosphate and the magnesium ion are shown using the dashed lines. And RAS is activated by the GDP exchange stimulated by the GEFS, and inactivated by the GTP hydrolysis stimulated by GAPS.
Discuss a specific mutation in this gene that leads to disrupts of cell cycle and may lead to uncontrolled cell division. The specific mutation that led to the disruption of the cell cycle and led to uncontrolled cell division is codon 12, 13, and 61. This mutation alters the GTPase function of RAS and interaction with the signaling of the molecule. It leads to the substitution of glycine with other amino acids such as alanine, G12, cysteine, G12C, aspartic acid, G12D, arginine, G12, and valine, G12V.